We'll have Tom Lowry and Elizabeth Erlach-Findel come up, please. Good afternoon. Thank you all for coming and presenting for us today. Our panel for today is Vaccines and Diagnostics. We have Thomas Lowry here from T2 Biosystems and Elizabeth Erlach-Findel from OIE, coming from a long distance. So we'll start with you, Thomas.
Thank you, Catherine. So my name is Tom Lowry. I'm the Chief Scientific Officer of T2 Biosystems, and today I'm going to talk to you about how a culture-independent diagnostic for bloodstream infections can help with the antimicrobial challenge and help get patients on effective therapy more quickly. So next slide, please. Just a brief introduction to T2 Biosystems. We were founded about 12 years ago out of MIT and MGH. And since then, we have invented, developed, and commercialized a technology we call T2 magnetic resonance. And this technology has allowed us now for the first time to have a battery of tests that can detect directly in whole blood the presence of pathogens and identify the species, identify the resistance markers, and report that out from a fully automated instrument in just a matter of a few hours. And these tests are now FDA cleared, and they're the only FDA cleared tests of this sort. There's been a lot of companies over the years that have tried to do this. I'm really excited that our technology can achieve that. I'm really grateful for the regulators at the FDA. They've been great to work with over the last 6 years to help get these tests out on the market. So go to the next slide. I wanted to focus a lot of my talk on sort of the paradigm right now of treating patients with bloodstream infections and how the diagnostic results and the therapeutic decisions depend on each other and what we're learning based on our experience. And I can hopefully circle around at the end to what I think can be done from your perspective to help in this area. So when a patient, or first off, sepsis, I think everyone's probably already familiar with. It's the most expensive hospital condition in the U.S. Where $20 billion are spent on this. And it's a paradigm that if you treat patients in this area, or if you've had a loved one or yourself have gone through this, it can become really staggering in terms of how the interplay of that diagnostic information and therapeutic, the antibiotic choice, can depend on each other. So one of the first things that occur when a patient's suspected of a bloodstream infection is that they have a blood draw that's taken. That blood draw then is put on a blood culture instrument, and the blood's incubated for a period of time to try to grow to see if there's any pathogen present. That takes quite a period of time, and on average it takes over a day, almost 2 days, to get a positive culture. During that time, patients are subjected to empiric therapy. It's basically an educated guess based on the epidemiology in the hospital, what the clinician's experience has been. And then as results come out of the lab, so results of, say, the gram stain result or the species ID result, or eventually resistance result, therapy is adjusted on the patient. Now, because of the risks of bloodstream infections, about 50 to 70% of all patients at risk of bloodstream infections are put on antibiotics, but really the positivity rate for blood culture is about 10%. So there's a significant overuse of antibiotics in the area of bloodstream infections. And additionally, those patients that are on antibiotics, almost half of them are not on effective therapy until you get down the line till you get that species identification and resistance result. So that having that species identification helps get over 90% of patients on effective therapy. If you go to the next slide, the reason why this matters for bloodstream infections is that every hour that goes by without effective therapy, the odds of death go up by 4%. So it's really critical that patients get put on an effective antibiotic therapy early and rapidly. The second reason is that actually the length of stay then, of course, is also tied to getting on effective therapy. And in fact, when patients undergo and reach the stage of septic shock, every hour that they're not on effective therapy reduces their survival likelihood by almost 8%. So it's really critical that these patients get on their effective therapy early. And one of the interesting paradigms is in oncology, we're very used to knowing exactly the type of cancer that a patient has when we put them on a therapy. But today in infectious diseases, that's not the case because the technology hasn't been available to actually identify those organisms and identify their resistance rapidly to help make those decisions rapidly. So if you go to the next slide. So the T2 magnetic resonance technology is the first culture-independent test that's been FDA cleared and is available for use in the U.S. here. The way the instrument works, basically the user takes a 4 ml EDTA blood vacutainer, loads it on the instrument, it's fully automated, sample to answer, and those results are reported out. So it's almost like a chemistry analyzer, but for ultra-high sensitivity, direct from blood detection of pathogens. So this instrument runs in a standard clinical microbiology laboratory. So we go to the next slide. So the paradigm for treating bloodstream infections for hospitals that have a T2 magnetic resonance instrument today is basically that they can obtain results much more rapidly and narrow that period of time over which patients are subjected to empiric therapy without any prior knowledge about whether or not they have an infection or what organism is infecting them. So within those first 3 to 5 hours, not only can patients have a species identification, but with our new panels that detect resistance markers, you can actually know if the patient is infected with a pathogen that hosts a resistant marker, like a MEK-A or KPC or whatever marker it might be. And that really represents an opportunity to have a paradigm shift in terms of how we treat these patients and how antibiotic stewardship is handled inside of a hospital. You can actually have information within the first few hours of the progression of disease to help impact that patient care. So once again, today empiric therapy is covering just over half, so almost half of patients are not effectively treated in those early time of therapy or early progression of disease. But then once they have species identification, you're above 90%, and then you get even higher once you know what resistance genes are present. So if you go to the next slide. So the panels that we have available today, we're continuing to expand those panels. The T2 bacteria panel was recently FDA cleared this last summer. It covers 90% of what are known as the escape pathogens. Those are pathogens that microbiologists consider often to escape the typical antimicrobial therapy. The T2 Canada panel is a panel that got FDA cleared about 4 years ago. Those cover 95% of fungal bloodstream pathogens. And then we have the Deputy Secretary mentioned Candida auris. We have a panel that detects Candida auris that the CDC's used to look at surveillance results of colonization of patients. And then we have a set of panels that have been funded and supported by Carbex. Carbex came up a little earlier. We're really grateful for our relationship with them and also the agencies that work directly with us through Carbex, BARDA, and the Wellcome Trust. They've been fantastic to support and give advice on these resistance panels. So we have, for the first time, direct from blood detection of resistance markers. So the shown here is the carboresistance plus. Those are the carbapenemase resistance genes. And our other panel covers the gram-positive resistance genes as well as the SPLs. So as we continue to expand these panels, we're seeing now the opportunity to really have an impact on patient therapy decisions early in the progression of disease. Next slide, please. So a lot of people ask me, well, what are the performance data for this type of test? Because it's a very different kind of test. It's a direct from blood. It's not using a positive blood culture. Like the term rapid diagnostics is typically reserved for those types of tests that are running off a positive culture. So the data that we have in hand show that, of course, test result is coming much more quickly, direct from blood, with just in a few hours, as opposed to almost 2 to 3 days, based on blood culture-based technologies. And then they can also allow for a much more rapid time to effective therapy, as some studies have shown for the T2 Canada panel. Sensitivities come in around 90%, north of 90%, specificities on the range of 98%. And then the published literature the KOLs are indicating that their NPVs and PPVs in different patient populations are very high, so that they can actually make antibiotic decisions based on the result. The T2 Canada panel has been out a little bit longer, for about 4 years, and there's some data now that have been shared showing significant cost savings, annualized cost savings for hospitals, as well as cost savings on a per tested basis, per patient basis, based on withholding antifungals. So we're really excited about now the building evidence that these interventions are really having an impact on patient care. Next slide, please. So one of the fascinating things as we participate in studies with different sites is with the T2 bacteria panel, we've asked a lot of questions about what therapies are patients on right in those first few hours of progression of disease. And our data are agreeing with the meta-analyses and other studies out there that you have a significant proportion of patients who are infected who are on the appropriate therapy for about 12 to 24 hours or even longer. And that's the opportunity to have this impact. Studies have shown that every hour of delayed effective therapy can cause a 2.7 hours increase in length of stay. And that if you get patients on that effective therapy in the first 6 to 12 hours, you can have a reduction in mortality of about 50%. The other fascinating dimension here is looking at stewardship opportunities. The studies that we've completed match also the studies that have been published there, is that patients are often on multiple unnecessary antibiotics. And so use of this kind of technology, I think, can help address the issue of overuse of antibiotics. Let's go to the next slide. So just in summary, my objective was just to describe the paradigm of bloodstream infection diagnostics and how it interplays with antimicrobial stewardship and and getting patients on effective therapy. That a direct from blood technology like this can really help aid in that decision-making process early in the progression of disease where it has a huge impact. I think now one of the challenges that we face is affecting adoption. Mel Weinstein's put it great at ID Week this last fall when he said the Holy Grail is direct from blood for sepsis diagnostics. Now we have different types of this Holy Grail here. How are we going to use it effectively? And that's a big area of discussion and one that we're very excited to be a part of, and we're excited to be a part of with different government groups and public groups as well. So thank you for your time.
Thank you very much. I look forward to the discussion on some of those issues of uptake and barriers.
Thank you to our panelists for enlightening us. I'd like to talk to Dr. Lowry about your technology. It looks very interesting and is certainly the way to go, or one really important way to go. I just want to ask you some technical questions about the limitations. So if you say it's good for about 10 common pathogens, and currently, and those 10 probably represent a very high percentage of true bacteremias, 90+%, I would guess. So if you get a positive signal, it's very helpful. But that if it's not a contaminant. And so the question is, how often do contaminants occur? What do you do in that case? And I'll just give you the second part of the question is, so that's what happens if you get a positive. Then if you get a negative, the sample still could be positive because there wasn't enough material for it to amplify, or it's not one of the pathogens that's in the panel of your 10. So what do you do then? And remembering that most blood cultures generally are negative. That would be the biggest group. So I'm interested in how you're proceeding.
Great questions. Those are common questions that we discuss with people, and that's part of this type of test is so different than your classic rapid diagnostic. So one of the interesting aspects of your two questions is the problem of culture negative sepsis. And there have been a few publications in that area, and there is a lot of interest for using a test like this to help address patient populations where it's hard to find if they're positive or not. Another aspect of your question is, as you mentioned, there's a coverage to our panels. Depending on the epidemiology in the hospital, the existing panels can cover around 2/3 to up to 90% of the pathogens, but it depends heavily on that epidemiology. And so use of these tests, and particularly because they don't represent all potential organisms, requires a strong stewardship committee. And so that they're using the result in the context of the different organisms that they expect to see. One of the compelling areas that we see is for like a Staph aureus, a positive or negative stewardship committee, looking at a patient population and making decisions based on that. It's really important, though, that results from this kind of test be used in patient populations knowing the risk profile. There are certain patients where de-escalations should not occur, but there's other patient population where antibiotics are being used, overused, where de-escalations can occur. I don't know if I addressed both your questions. I can circle back to the first one if you'd like.
That's very helpful, clearly. The question about contamination, because Staph aureus can be a contaminant also. Staph epi can be a contaminant or a real pathogen. Yep. So how does your test enable the clinician to better interpret the results?
So specifically for Staph epi, that's not a reported result, and that doesn't cross-react with our results. So that was an important design consideration in these panels. Mm-hmm. But the general concept of when I get a positive on T2 and I have a negative on blood culture, what do I do? Because that could be a culture negative sepsis or it could be a contaminant. And that's part of the publications that are building in terms of understanding how to do that. A lot of times stewardship committees react by saying, well, if I have a positive for Pseudomonas, I'm already running the culture, I've already got the patient on therapies, I'm going to adjust the therapy to keep them covered because the likelihood of infection is higher because of that Pseudomonas positive. So there's a different kind of thinking here in terms of risk profiling the patients with a test like this to help inform those decisions early on that right now are just based on very, very little information.
Yeah. Congratulations. Sounds good.
Thanks.
David?
Thank you. A question for Thomas. Rapid culture independent diagnostics definitely are desirable, but have you talked much about the impact of this, what's going to happen with surveillance? I think CDC is struggling with culture-independent diagnostics for many of the surveillance programs. And,
Yeah.
How do we look at the benefits of both in the future?
Yeah, I think what's really important there is that culture-independent diagnostics, they really need to look at their attributes different than blood culture itself. And in the community, we're hearing loud and clear that a culture-confirmed case, oftentimes they treat differently for surveillance than a culture-independent case clinically as well. But right now, they're binned together, and that can create challenges for hospitals and disincentives that cause them not to want to know. And so, one of the opportunities here is you have a test that doesn't have the shortcomings that culture has in terms of, and for example, our data show that one tube from our test takes 3 cultures to get that positive. And so you have a test that's catching positives more quickly. Do you want to disincentivize use of a test like that to capture infections early by the surveillance? I think that's an active area of discussion that's important to hash out for these types of tests in particular. And it's very tied to bloodstream infections in this case, and that's different than the other types of rapid direct from sample tests.
So, Tom, I find T2 very interesting and congratulate you. 5-7 years ago, if you would have told people that there was going to be an assay out there directly from blood that could detect down to 1 to 3 colony-forming units per mil, there would have been much rejoicing. People would have been totally wowed by that. And now we have this test, and the uptake is quite limited. And my thinking on this is it's because clinicians really struggle with incomplete information. So you have an identification of only the pathogens in the panel. You have a sensitivity that for some pathogens is in the 90% range, so a negative result you're not sure of. You do have issues where you get positives that you can't confirm by other methods. And this is all the growing pains around adapting to new technology. Can you give us some insights into T2's approach to how to help clinicians deal with incomplete information, how to help clinicians deal with new technologies that come out that aren't exactly like the ones that they're comfortable with?
That's a great question, Angie. Thanks. I'll sort of go 2 parts. One is one of the active things we do is we have a group of medical science liaisons that are previously on antimicrobial stewardships committees. They're PharmDs. And they engage on a hospital-by-hospital basis to discuss how a test like this could be implemented effectively in a hospital's given patient population. I think that the question of if we had all 100+ pathogens that all of the microbiologists would love to see on there, which it will happen, and it's going to. But if we had that today, what would uptake look like? I think a test that's direct from blood represents a new paradigm in terms of the processes in a hospital, how a laboratory works with the microbiology, with the stewardship committee and with infection control. And there's all these different misaligned pressures and incentives, and this technology represents a riddle. And so hospitals that have groups, those groups that work really well together and sort of see the vision of what this promise is, those are the hospitals that we have good success with. But hospitals where stewardship committees aren't really fully implemented, or there's a silo mentality when it comes to budgets and other things, they're the ones that it takes a long time to sort of embrace that. We do get universally generally the response, hey, that's awesome, I've been waiting for that. And it's like, well, how do I implement that in my institution? And a lot of times laboratories are wired to bring in something that they already have a line item in their budget for. But fortunately, there's been a fair number of hospitals that want to work through those barriers, but it's those ones that work best together across those disciplines. Does that help address the question?
Bob, did you still have a question? Okay. Christine?
I'd like to thank the panelists for their excellent talks, and this question is for you, Tom. I highly applaud T2 for being very innovative and the first in the field in direct sepsis in this way. I think it brings a new level of diagnostics to the field. But there are, as Angie mentioned, a few issues, and one of the big ones is that we have with this type of technology, maybe not so much for Candida because you have a very defined patient population, but for the general person coming in through the emergency room where, say, they're ruling out a bacteremia, it doesn't even have to be sepsis. It could be just simply a bacteremia. where we have a positivity rate of 10%, but yet the other 90%. So we're doing a tremendous number of blood cultures, of course, a lot used inappropriately, but how do we select that actual patient population that's going to benefit from this? We know it's a good test, but it's a very expensive test. You're limited by the number of tests you can do in a certain time frame by instrumentation. So that's one question that I have about, is it clinical algorithms where we use other biomarkers maybe to help enrich or select for the patient most apt to have a positive T2? I mean, we did 300,000 blood cultures a year in my lab. I could not run a T2 on every patient coming in that would have a blood culture. So I think defining those algorithms and how you work to help with that. And the other thing is that we talked about carve-outs. So for new therapies, whether they're immunomodulatory therapies or, new antibiotics that are going to be very expensive. But yet, under a DRG, every time the test is worth it in the cases where they're positive, we know it saves tremendous amount of money overall in the healthcare system, but it's a huge also financial burden. So how can we go about in a recommendation where critical tests like this should be maybe some type of a carve-out, not part of an actual DRG, so that we can use it in the right patient population, making sure we select the right patients, but then not being penalized because of costs and other things for that. So has T2 thought about that as far as approaching reimbursement and payers for this type of carve-outs?
Yes, I'd be happy to go through those. I think I captured about 4 or 5, but I probably missed another half dozen. But maybe if I could ask, you said it's an expensive test. Could you define what that means from what you've heard?
I don't mean expensive from a clinical or an outcome standpoint. Yeah. I think it's a cheap test when you get a positive answer and you can quickly make a decision and reduce antibiotic use or more appropriately select it. Unfortunately, we live in very siloed worlds, particularly within the hospital setting. So it's an expensive test for the laboratory. I'm not talking about clinical benefit. I believe in economic outcome studies and whatever. And
do you have a dollar number that you have in mind when you say that?
No, it's all relative. For me, I don't think it's an expensive test because I know the value of it. But if you're going to add $2 or $3 million on your laboratory budget to bring in a new test, we have to justify what that would be. Yeah. So in the administrative, the bean counter world, some of these things are looked at as expensive tests that we have to show the clinical benefit of it. Okay, we know it, but we have to prove what that clinical benefit is. So that's where health outcome studies become extremely important, like you've done with Candida. So that's how I reference it as expensive compared to 10-cent glucose or, not the same clinical impact, but they look at these things differently when you're looking at a hospital budget. So when we have these type of tests, how could we approach it, do you think, so that maybe these type of highly clinically impact tests would be a carve-out, would not be part of that, say, DRG of that patient coming in?
Yeah, the reason why I asked, there's a common misconception about our tests that they're highly expensive tests. We're really priced at or below the level of any rapid diagnostic out there. Mm-hmm. But you do raise a very valid point about how do you select patients. Mm-hmm. I think everyone would agree if you had a test that was comprehensive and you could resolve budget issues, whatever those be, I mean, even if you had 10-cent test, it's going to be a lot of money in a budget. This would be a huge value add to clinical care. And so I think the question becomes, as we go into hospitals, we're never proposing to add $2 to $3 million to their budget, absolutely not. But it's looking at their patient populations, and whether it's in the ED or in other places, helping them select those populations where they're looking at a couple $100,000 the first year, or a few $100,000 the first year, in order for them to then evaluate if they see that real value. And you alluded to correctly, this all falls underneath the DRG. And so the hospital's having to then do an analysis to say, are they going to see an economic benefit? Of course, at the same time, they want to do an analysis to show that they see a clinical benefit in their patients. And I think those data will build so that hospitals won't have to spend as much time do that themselves. But that's one of the burdens they face, bringing a very new technology, as they have to prove that out themselves. And we typically engage on a hospital-by-hospital basis and help partner with them to help them achieve those goals in a way that's cost neutral to them or is not financially painful to them. We are, working with CMS is an important thing to do. And there's programs like the NTAP that can help there. But I think earlier there was the concept brought up that for, I think it was for antimicrobials as well as vaccines, could they be outside of the DRG and have their own code? I think that could help tremendously for situations like this. But I think use of something like this does need to be tied to a stewardship program, and that does need to be tied to certain things that a hospital should have so that they can actually realize the efficacy of test results like this. And I would just circle back. I've got a few other ones that you brought up that we have thought about each one. So throughput, one of our instruments can run about 5,000 high-risk tests patients a year. There are institutions that bring in multiple instruments, and there's a higher throughput version of our instrument that could be developed as well. And then you bring up, and it's not uncommon, so what do I do when I detect something that culture missed? And it's a fascinating paradigm because, like, well, you told me you wanted the Holy Grail. You detect something. Now, certainly, are there ones that are hard to prove out? Yes. Have we gone to great extents trying to look at other sources for the infection, using other technologies to detect that and try to sort that out? And that's where I think, actually, if we had brought the expansive panel to the market right out of the gate, the conversation would be very different. Be like, I'm overwhelmed with all this new information. I don't know what to do with it. And so what we're seeing is that hospitals are working through this new paradigm with these limited panels, and then we're continuing to expand that panel to where we have a comprehensive solution. I think will be a great impact. I think what could be really helpful is pull incentives not only on the cost side, but also in terms of helping demonstrate the clinical populations where technology like this can have the biggest impact. And then maybe it's tying certain incentives to those populations. But right now, ourselves and hospitals are having to partner to help push that forward.
Okay, thank you.
Dennis?
Thank you. I want to belabor a point that was initiated by Angie and then Christine picked up on. And so while I'm repeating other people, I think it's such a nuanced issue and so significant it does bear repeating. That is the need for algorithms to target the population and to educate people on how to interpret the results. And so Neil Clancy gives some very nice internet presentations on T2 in Canada, making the point and showing how which population you use it in where you get benefit and which ones you use it in where you don't get benefit. So I think the education and our outreach, not only to the experts who are going to use it in the diagnostics lab, but to the people who are going to be ordering the test and then making a decision based on it. And so, Marty, you liked the analogy this morning about the GPS and the airplane. I'll probably fail on this one, but another example might be a scalpel is a perfectly wonderful tool, best wielded by a surgeon, but not on everybody that walks through their door.
If I could just respond, I totally agree with that, and that's why we have the MSL program that we have. So every hospital we try to engage to find that targeted population where benefit could most likely be realized, and then we encourage them to publish and share that data. Neil's a great example of that kind of collaboration.
And I second the congratulations on your company making a major stride in taking this forward, and I think it's a step in the pathway, so that's why I was making these comments, is how do we move to the next step without pushing too quickly so that companies that preceded this one don't fall by the wayside. Gorham Lisby gave this concept at an international workshop we did on diagnostics that several predecessor devices that failed didn't fail because of the technology, but because of the lack of algorithms to guide their use.
Yeah, one of the fascinating experiences I've had, and Angie referenced this a little earlier, over the last 12 years, is when we first chose to go into this area, the first response we got was, that's not possible because so many people have tried to get to that 1 to 3 CFU per ml. Once we demonstrated that, then the response was, well, you're not going to be able to get regulatory clearance because so many companies have tried to clear that bar of excellence that should be there for a test like this. And then once we've gotten past that, now it's all right, now you got to prove to us where to use this kind of technology effectively. And we're committed as a company to try to see our way through that. But we really welcome partnering, whether it's in the private sector with hospitals or with government agencies, to unpack this riddle, because I think we have a big opportunity here, but it's going to take a collective effort to do it in a way that's expeditious and has appropriate stewardship of the test as well as antibiotics.
Helen?
Really quick follow-up. Coming back to the stewardship and just thinking about the potential benefits, not to an individual patient, but to all our patients from having these results. So the idea that knowing sooner that there's an organism that could be resistant and implementing the appropriate infection control measures can make a huge difference, not necessarily for that patient, but for all our patients. And I'm just curious if you've done any work on this in terms of trying to quantify those kind of advantages, and has it had any traction with people you're doing business with?
Yes, I'm glad you brought this up. We have a paper, Hong Nguyen is the lead author on our pivotal study paper, and that's in final stages of review, that talks a little about this, and she's presented this at ECCMID last year. And then another paper that's in queue as well, where we went in and asked the question in our pivotal study, what was the antibiotic utilization in that patient population? One of the fascinating things to what you're pointing out is that 1,427 patients in total were in the study, and over 60% of them were on an antibiotic that would be effective against the 5 species on our panel. And then we asked the question, well, of the ones who were actually infected and identified by blood culture, that would be 35, what was their coverage? And only 20% were on effective therapy. And so there's a lot of dimensions of complexity in that data set. I won't initially go into it, but just reinforce this concept that we're using antibiotics very aggressively because of the dire situation of these patients, but we're sort of shooting in the dark. So I think there's a huge benefit to help protect the antibiotics we have by having informed stewardship decisions a lot earlier.
This question is for Tom. Thank you very much for both of your presentations this afternoon. Unless I missed it, what is the potential, and what do you anticipate a rollout would be for use of this technology in a low and middle income country? I think it's great. It's absolutely spectacular to have this type of technology available. But really, when we talk about the emergence of MDR pathogens and those that concern us the most, they're going to come from countries that likely cannot afford this type of technology, or there would be a problem in keeping the kits in supply and various other aspects of all of that. So how can you see rolling this out to those countries sooner rather than later?
Maybe 2 answers to that. The first one is not characterizing any country that I'll refer to as low and middle income, because I'm not an expert in that categorization. But we do have a presence ex-US, and what's been fascinating in southeastern Europe, where you have really high prevalence rates, there's been a lot of enthusiasm, particularly about the carbapenemase panel that we have there, but being able to use this kind of technology. And the kind of partnerships that I see working there, that we have a distributor model there, and so they work out the finances between them. And I think when it comes to that more broadly, I'm not an expert in that. I imagine there may be some learnings from the Gates Foundation and with the rollout of the TB tests. For our technology, we do need a laboratory that has power and has environmental control in it, and we do need a reliable shipping process. But we are in the Middle East and Southeastern Europe as well.
Okay, Angie, and then Lonnie will wrap us up.
So, Tom, I was wondering if you could give us a perspective on how you funded the company, to give us kind of an idea of how do diagnostics development get funded?
Yeah. I have to say, we've had a history of incredible investors, VCs with hearts, and many other investors. We are public now, who've really believed in the long-term vision of what we're trying to achieve. So we were privately financed until 2014 when we went public. Now we live the bumpy and bruisey life of being a public company, but it's we have a great base of investors right now as well. We've had non-dilutive funding along the way that's been helpful, like CARB-X, as well as some other non-dilutive funding early on in the company. So that's also been a big help. Non-dilutive funding makes a huge difference for a company like ourselves, because we're trying to be a commercial company while still investing heavily in development and expanding our pipeline. And so really, all of our investors we're greatly appreciative for. They certainly have had a high bar of excellence that they've held us to, and we've been happy to be able to meet their requirements over the years.
The thought of a new test is really fascinating, and I sit here caught up myself in the excitement about determining the correct population to apply it to and interpreting it within different populations. I just have the question for the council, whether we have that same fervor in making sure our other diagnostic tests are as appropriately applied and interpreted. And it's just like getting excited about new antibiotics, but not going back and reevaluating our use, duration, dose, et cetera, for our previous ones. And I think this discussion, I hope for the council, stimulates the thought that maybe as we look at the next 5 years, one of the plans is doubling down again on appropriate test selection, interpretation in light of the specific population it's being applied in, and also, again, revisiting our therapeutic strategies using the tools we have. So that, I'm excited too. I think it's really neat stuff, but I want us to use that fervor to go back and double down on the tests and the tools we have today too.
And on that, thank you very much. Thank you, Elizabeth and Tom, for your time and sharing your information with the committee. Please join me in thanking our panelists.